Antibiotic resistance is, of course, a bacterial problem, because in every bacterial population we have billions of cells, and the bacterial DNA mutation frequency is really, really high. And also because we have in the bacteria genome an haploid genome, so no diploid genome to contrast the mutation. In this situation, we can have the very high mutation strains, so the hypermutable strains, that are strains that lacks the protein set to repair mutation bias. And these strains can have a benefit when the antibiotic pressure is high. We can divide the emergence evolution into two different periods. The pre-antibiotics period, a very big period of billions of years called macroevolution, without antibiotic use. In this period, environmental factors stress some existing mechanisms of bacteria with survival function, resulting in non-selective mechanisms, like, for example, efflux pumps, the pump for detoxification of big genome bacteria that are typical of Pseudomonas and Acinetobacter, for example. And we have the post-antibiotics period, so the period in which we use the massive use of antibiotics. This is a very, very short period of 70 years only. And in this period, the anthropogenic factors and the massive use of antibiotics determine the really growing of resistance mechanisms, and these are acquired mechanisms. And in this period, we also see that we have the shift among opportunistic bacteria into pathogenic bacteria, like Pseudomonas, for example. And actually, we can call these bacteria like superbugs. And about AMR mechanisms, one of the most important that is possible among gram-negative bacteria is, for example, the conjugation. The conjugation is also possible among different genera and species. And we actually know that, for example, in the intestinal microbiota, the E. coli, that is a normal intestinal colonizers and opportunistic bacteria, can share resistant genes with other pathogenic bacteria, pathogenic species like Salmonella species, resulting into very difficult salmonellosis disease to treat because of few treatments available. And about the acquired mechanisms, I can show you, for example, the mobile genetic elements. These are the elements, genetic elements that are specialized into capture genes encoding for example, for AMR resistance, and they can move anywhere in the genome, these genes, like integron, for example, or transposons. These are jumping genes or plasmids. But we actually know from the literature that the AMR phenomenon is constantly increasing, and it is predictable in case of incorrect use, for example. During an incorrect use of antibiotics, the onset of AMR, it can be really, really fast, like 7 days. But we also know that the AMR phenomenon is developing into cross-resistance, for example. The cross-resistance is the resistance against more antibiotic, but in the same categories. And this phenomenon can turn into the most important and the most dangerous, of course, multi-drug resistance. That is the resistance against more antibiotic categories. A dramatic example of cross-resistance that can turn into multi-drug resistance is the methicillin resistant of Staphylococcus aureus and Staphylococcus pseudointermedius. These are both coagulase-positive staphylococci, so pathogen staphylococci, and they can share the meca gene, for example, with coagulase-negative staphylococci, so opportunistic staphylococci. Meca gene is a gene enclosed in isochichimic acid, codifying for unaltered penicillin-binding proteins, resulting into lower affinity for beta-lactamase and clinically in resistance against all beta-lactams. But actually, we know that these pathogenic bacteria, methicillin-resistant staphylococci, they can be resistant also against other antimicrobial categories. And for example, in human medicine, we have a lot of report about this, about MRSA, methicillin-resistant Staphylococcus aureus, that can associate the meca gene with other resistant genes and resulting into resistance against other categories with very, very high percentage resistance rates. And we demonstrate more or less the same in veterinary medicine because in cooperation with the University of Cambridge, my research group demonstrate that also, for example, for Staphylococcus pseudointermedius, these are typical staphylococci, pathogen staphylococci for animals, they are much more resistant against other categories. We sampled 68 clinical isolates of Staphylococcus pseudointermedius coming from canine pyoderma, deep canine pyoderma, and we tested these staphylococci, these strains, for the presence of MECA gene with the PCR. And we found the 31% positivity, so 31% of strains were positive for MECA gene, so they are methicillin-resistant Staphylococcus pseudointermedius. But the most important things that I want to show you is that when we compared the MRSP, so methicillin-resistant strains, with the MSSP, the methicillin-susceptible strains, we found that MRSP were much more resistant against other antimicrobial categories and not only beta-lactams. And another example of cross-resistance that can turn into multi-drug resistance is the extended spectrum beta-lactamase detection of gram-negative. The extended spectrum beta-lactamase are a group of enzymes, and this group of enzymes is codified from a family of genes. And this enzyme can hydrolyze the beta-lactam rings of the beta-lactams antibiotics, of course, reducing the activity of beta-lactams antibiotics. And of course, like the same things that we demonstrate for staphylococci and that we have in staphylococci, we have also for gram-negative SABS bacteria, because these bacteria are, it is demonstrated that they are much more resistant also against other antimicrobial categories. So the multi-drug resistance is the acquired resistance and against more than 3 antimicrobial categories by storing of different genes and different mechanisms. For example, multi-drug efflux pumps, integrons, MECA gene, all together. The only successful approach to these dangerous problems is the One Health approach, because we have to consider that bacteria population can share genes. So humans, animals, and environment, they can share resistant genes. I said in a previous slide that the AMR is predictable in case of antibiotic use. But we actually know also that this phenomenon is increasing also in wild environment, and especially in aquatic environment. And then we can use some marine species as AMR, they can be considered as AMR reservoir. This is an ecological problem because AMR bacteria found in marine environment, they are an index of marine pollution, and we have to by monitoring this trend. This is the poster that my research group present in the 6th Mediterranean Conference on Marine Turtles in Porridge last year about multi-drug resistance in clinical isolate from loggerhead sea turtles living in the Mediterranean Sea. But why we can consider the Caretta caretta as sentinels of our sea? Caretta caretta species are really good bioindicators since they have no history of therapeutic antibiotic exposure. They have long lifespan, a long period to reach sex maturity, and they feed near to the coast, especially in winter season. They are also one of the species enclosed in the REST list, the list of the World Conservation Union, because of the decreasing number of these species, and not only in the Mediterranean Sea, but worldwide. In the Department of Veterinary Medicine, of course, you know, we have the sea turtle clinics, that is a rehabilitation center for Caretta caretta, and my bacteriological lab cooperates with the sea turtle clinic in order to investigate bacteriological wounds, for example, of Caretta caretta, because for wild and non-conventional species like Caretta caretta, we have few pharmacogenetics and pharmacodynamic studies, and we have few even known registered molecules. So it's really important to avoid the MR developed in this species because we have really a few treatments available at least. In a study period of 2 years, We sampled 52 injured caretta coming from Puglia, the Adriatic Sea, and the source of samples were for 32% BAL, so samples from respiratory tract, and 21 from biopsy, and 15 from carapax wound and skin wounds, and then auricular swabs, cloacal swabs, and really few percentage of plastron wound. We performed the bacteria cultivation of these lesions, and of course the bacteria identification, and the antibiotic-resistant profile, and the SABS gene screening. The most important things that I want to point is that no antibiotic treatment was done on these animals before this sampling. We received in the Sitarto clinic more or less 400 caretta per year, and especially these animals arrived in the winter season, because in winter season we have the unfortunately mixing among caretta, and fishermen. In winter season we received more or less 6-7 turtles per day, and most of them for traveling, for accidentally catching during traveling. So when they arrived in the center, in most of the cases they are injured because of traveling. So they were subjected to, when they arrived in the clinic, they were subjected to physical and behavioral trials, all the measurements, and or the external examination for wounds, for example. For the bacteria cultivation, we used 3 different agar media, the Columbia blue agar, Mekong agar, and Triptysoy agar, and we cultivate the bacteria into aerobical condition for at least 2 days in 37 degrees. We used for the identification of bacteria phenotypical and biochemical tests. Antibiotic screening was done using Kirby-Bauer methods, and the molecules tested were, as you can see, penicillins, cephalosporins, and carbapenems, so different classes of beta-lactams, and then other categories of antibiotics, fluoroquinolones, tetracyclines, and glycosamides. Among the big family of SABS genes, we decided to screening only 4 of them because they are the most representative and the most investigated genes: MPC, ACTXM, TEM, and SHV. After the PCR, we did the enzymatic purification and the sequence analysis. We analyzed the sequence analysis with Genius and 2 genes database, the BLAST database and ResFinder that is specific for SABS gene. And these are the results. In total, we correctly identified 40 gram-negative strains, and 55% coming from cutaneous wound, 32.5 from BAL, and 12.5 from internal lesion. And the most representative general percentage, as you can see, are Aeromonas, Citrobacter, Vibrio, and Pseudomonas, followed by Proteus, Acinetobacter, Morganella, and really few percentage of Klebsiella, Enterobacter, and Narcolepsans. But the most important things that I want to show you is the resistance rate, because we found a very high resistance rate against all the categories that we tested, not only beta-lactams, but the resistance rate against beta-lactams was really unexpected and really high, because as you can see, against imipenem, but for ampicillin is more or less the same, we reported 100% of resistance. So all the strains tested were resistant against imipenem, that is a carbapenems. And 97.5 against ampicillin, 90 against amoxiclavulanic acid, and 90 the same for cephalexin, and 77.5 against cefotaxime, and 72.5 against cefuroxime. So all the beta-lactams were the bacteria were very high resistant against beta-lactams, but also against other categories, for example, fluoroquinolones. And this is an example that I want to, of course, I want to present to you because 9 out of 40 of these strains, so 22% of strains, were found resistant to all the molecules tested. As in this case, we found 2 genera in this BAL from Acaretta caretta, Daphne, and we found Acitrobacter and an Aeromonas, and these 2 genera, these 2 bacteria, were found resistant to all the molecules tested. Because we found 87.9% of strains showing resistance against at least one beta-lactams tested, we decided to screen for SABL's gene. And these are the results. We found that 20 out of 40 strains, so 50% of these strains, were found positive for at least one gene. The most important, the most found was MPC for 45%, and then CTXM, 17.5%, followed by TEM and SHV. And we found also two genes together in 17.5% of strains, and 3 genes together in 5% of the strains. And these are the complicated results from BLAST database because, for example, for SHV is an extended spectrum beta-lactamase found in Escherichia fergusoni, and the BLAST-CTXM is a gene for class A extended spectrum beta-lactamase found in Enterobacter cobae. And TEM is a gene for class A extended spectrum beta-lactamase found in E. coli. And from the MPC group of genes, because MPC is a very big fragment, so we found different genes. We have 3 of them that are cephalosporins, hydrolyzing class C beta-lactamase found in Enterobacter spp, and 1 found in Citrobacter freundi, and 1 in Morganella morgani. So when I summarize all the studies about marine animals that use the culture methods, the methods that we used for our study, I realized that we have few research studies about Caretta caretta, because a lot of studies involved other marine animals, like Chelonia is one of the most used for this type of research, and for example, sea mammals or seabirds. So we have few studies about Caretta caretta species, and we have also a few reports about clinical isolates, and of course also about AMR evaluation. We have also we have these results because we have few reports about injured turtles. And about the general percentage, we didn't report the presence of, for example, E. coli or Salmonella species that are considered index of fecal contamination, and they are, of course, possible in Caretta caretta wounds, but we didn't have this type of results. And we can compare our results with the other result that I previously showed, because most of the other studies report like Citrobacter and Pseudomonas as major findings, and we can compare our result with this one. But both marine turtles report and marine mammals report, they considered bacteria with a secondary role to, for example, parasites or viruses, because we don't know, for example, what is the role of these bacteria in these wounds. We don't have studies about the comparison healthy tissue or injured tissue. So we have to understand what is the role of these bacteria, if they are opportunistic pathogens or pathogens, just pathogens. We found 50% of the strains, as I said, at least positive for one SABL's gene. And this is a really interesting data because we don't have report about SABLs in Caretta caretta. We have just one study about this. It was 10 years ago, more or less, in the same area, in the same Mediterranean area. They screened with a phenotypical test. They screened the isolates. And because they didn't find any positivity, they didn't use any molecular characterization, any molecular investigation. So we found, instead we found 50% of strain positive. So it's completely different, the results. And about the genes, I summarized more or less all the information about these 4 genes, because these are resistance genes that are involved in the antibiotic inactivation. They are responsible for the inactivation especially of beta-lactams, carbapenems, and cephalosporins classes. And the most important thing is that these genes have human origins. They have human, urine, and fishes origins. But we found a very high resistance rate. So probably for the future, we have to screen other SABLs gene, not screened at the moment. Or probably it will be interesting to understand also other mechanisms, other resistance mechanisms, like porting expression on efflux pumps system, in order to understand what is the origins and what are the mechanisms of multi-drug resistance rates, like the one that I show you. And about DiMeV, it's really important to talk about these molecules, because this is a carbapenems, as I said, it is a beta-lactams licensed only for human use and only for hospitalized person, so not for animals. And we reported 100% of resistance against this antibiotic. We don't have in the literature any reports about this really high resistance percentage, and the only one report that I can cite is against 40 of 10 years ago. They screened 31 molecules, and they reported the lowest resistance of imipenem among 31 molecules tested. So our results in 10 years, I mean, the situation is completely changed, and because our results are completely the opposite. And about fluoroquinolones, we tested fluoroquinolones of new generation, and the one that is only for veterinary use only, you know, the aerofloxacin is just for animals, and the other ciprofloxacin, moxifloxacin, and pradofloxacin that are fluoroquinolones only for human use only. And again, we found a very high resistance. So it's important to understand what are the mechanisms involved in this really, really high percentage of resistance. So for the future, it will be interesting to understand and to analyze other mechanisms. Of course, the Tartu-Biome project is the project, as Sunchiza said, about the normal epizoic and endozoic microflora. But this project can help us to understand also what are the origins and what are the mechanisms of this bacteria, because it's really important also to compare the damaged tissue, the injured tissue, with undamaged tissue in order to understand what are the role of this bacteria. And of course, we want to investigate more about AMR resistance mechanisms found in marine species. So we have the last report about EFSA in February of this year that showed that antimicrobial resistance is showing no signs of slowing down. And in humans medicine, actually, we have still few reports about pan-antibiotic resistant bacteria, but at least we have. And this is a very, dangerous problem and growing. And I can conclude that our SABLs genes, the genes that we found in Caretta caretta, they have a human's origin. So, of course, this is an index of marine contamination from humans. But in most of the cases, because we are a vet, the question is to treat or not to treat these animals. And of course, clinician can confirm this, with systemic antibiotic. My colleagues, my PhD colleagues, Erika Grandolfo, in the cooperation with Cambridge, they demonstrate in MRSA and MRSP that more antibiotic resistance genes bacteria have, less virulence factor they have. So in some cases, when you found an antibiotic-resistant bacteria, this bacteria is less virulent. It is really interesting also because antibiotic resistance is not a virulence factor, it's a survival factor. And we don't have report about these findings on gram-negative, so for the future, it will be interesting also investigate on these things, but for gram-negative bacteria. In a lot of cases, Caretta caretta in the rescue center were treated with systemic antibiotics, only if it is strictly necessary. because they tend to use topic natural substance, and Professor Franchini will discuss with you about this new treatment. But we considered the MR screening really mandatory, not only for Caretta caretta, but for all species and also humans. But for Caretta caretta, in this case, this type of test is really mandatory in order to choose what is the best treatment, and also because this is an endangered species and we have to preserve it. I want to thank again Professor Junsi-Jambosek for her lecture, and the head of the Sea Turtle Clinic, Professor Di Bello, and all the staff, all master students, and all my colleagues. Of course, Maria Laura Corrente, my tutor of infectious diseases of department, and also Pasquale Salvemini of the WWF Molfetta, and my colleagues that is not here at the moment, Giorgia Diacudi for biomolecular investigation, and all of you for your attention.
Thank you very much, Adriana. As you can see, Adriana is also an agar art artist, yeah. So he finds time to joke with bacteria. Yeah. But it's not a matter of joke, as you can see. I remember when we first found the pattern of antibiotic resistance. This is not from wild animals, but from hospital-acquired infection of humans. Is possible, so we have to study deeply this phenomenon.
Of course.
Okay, thank you. So is the time for a discussion. Any question? For Sanchika or Adriana? Also in Italian, please.
No, I have just a curiosity for Sanchika. Because you said that we have a lot of studies about gut microbiota and yes. Few studies about skin microbiota.
Yes,
why?
A few.
In your opinion, because it's really important to investigate gut more than...
Gut, because this is connected with the food and with how to treat animals, not for, like you do, the wounds and the infections. It's more connected. If we know what resides in the gut, we know what to feed them. And it's influenced on the husbandry of the turtles themselves. So, and it's also much easier probably to get the results, I have to say, because...
But, I mean, it's really interesting also to investigate the skin, because the skin is the surface and the contact with the external
From sea turtles, I'm not aware of any study that is published on the skin composition of the sea turtles, any of the species.
So yours will be the first.
The pioneer. I think if maybe somebody is working somewhere in the world, but not that I'm aware of.
No studies, published yet. Okay, thank you.
So it could be another interesting point of research for us. Maybe.
Yes, the point is in this project, everything, like every day there is something, okay, this is novel, this is new, I don't know, there is no study on it, okay, what to do with it. So it is the constant path of discoveries, but we need to pick certain topics that are interested, and I think really with the connection with the research, with the wounds, and it's really important thing to study, really, and I was completely overwhelmed by your results from the antibiotic-resistant. It's like I don't think that anybody is expecting that the bacteria taken from the wound on the head of the sea turtle have 100% of resistance to the human antibiotics. So
this is an important results. I mean, really, I was afraid when I was for the first time some antibiotic-resistant bacteria.
Any further question?
For Sanchika, very, very interesting your lecture. Thank you. When you say every day is a new day, for us today, every day is the same, because we have a lot of turtles, as you know. We have almost 300 turtles for years, so every day we have a lot of new pathology, new cases, and turtles is not like other animals.
Yes.
This is the first thing that I say to the students, because turtles are different from mammals, but are different from other reptilians,
Yes.
very different, also for the environment where they live. So my question is, in your opinion, diatomae are pathogen?
No.
No, okay.
No, they the biofilm that is forming, it's when the pathogeny can be, you know, not pathogeny, not pathogenic, but more like let's say nuisance.
Okay.
for the animals. For instance, when it's not just the diatoms, the caretta, it's normal for her to have a biofilm. But when it's injured, when it's a bad state, it has more. It's like your teeth. I mean, the biofilm is formed on your teeth. When you don't brush it, it can, you know, extend it to caries and so on. You need to clean it regularly, but it's not pathogenic per se.
Thank you. My question is because in cytology of Ball, we find that when always when the turtles are drowning.
Yes.
Yeah.
Yes,
we found the algae and the diatoms.
And the diatoms, yes. This is from but it's the diatoms are then the origin of the diatoms is from the seawater.
Yeah.
So it's not the ones that live with their environment. So it's I mean, diatoms are usually used as a, let's say, indicator of the drowning.
Of the drowning, yeah. We use it as an indicator of the drowning, yeah. Another thing for Adriana and you, no.
Of course.
We are no rehabilitation center, we are a clinic. It's
okay,
different.
sorry.
No, that's one. Because we have only sick animals, and then we
move the animals to the rehabilitation,
center, yeah.
Okay.
WWF and so on.
Yeah.
Another question? We have a few minutes for another question. Nobody? Okay, so I can invite Delia for her lecture. Just 1 minute to prepare. I can thank Antonio Chiaravalle for his technical support. Every conference without Antonio is not possible. And also the bar, because as you know, the University of Bari is plastic free, so we have glass.
It's good.
It's good. We try to be plastic free. Yes, but Delia has. But you can recycle the bottle, Delia. Sorry? You can recycle the bottle.
It's not a mono-use bottle.
Yeah, the sea turtles are like this flagship species for the plastic straws, for instance. You know, the worst thing that you can see is the turtle with a straw from the nostrils. It's like, but it really has an impact. I mean, we've been in several involved with in Pula, and also in our university, in the department, we had this scientific open days, and you influence on kids definitely with the sea turtles and plastic, and you see their change of attitude, like, oh, mom, I don't want a plastic straw because the turtles are threatened by it. So it's a good thing.
Yeah, it's important because kids are more susceptible than ours.
And then they persuade their parents not to use straws and plastic.
I'm optimistic because young people are more involved in ecological problems. You know, Greta Thunberg. So we can hope in the future.
Greta è stata bocciata.
Greta è stata bocciata.
Oh, really bad information arrived from Stefano. Greta was blocked because of the lot of days out of the school. So.
Okay, Delia, for your
you.
amletic question, another amletic question, to
Oh,
treat
yeah.
or not, to amputate or not.
Yeah.
Okay.
It's a really, really pleasure for me to be here for this kind of meeting, because the sea turtle now are the major thing that we study in this time. So I thank Maria Laura for this organization. I thank Sanchisa for to be here. Today I want to talk about entanglement. Entanglement is in sea turtles, perhaps in my opinion, is the most serious problem for sea turtle, worse than ingestion of plastic, the worst serious problem for pollution, of course. Because caretta are carnivores, are not herbivores, not as Chelonia midas. Chelonia midas is herbivores. Caretta does ingest much plastic, but entanglement is a very, very, very big problem. Plastic in the seas is a dramatically increasing problem and has been described as a serious aspect of pollution. Marine turtle entanglement is defined as the process under which a marine turtle becomes entwined or trapped within anthropogenic material. Some of the most affected animals include cetacean, pinnipeds, seabirds, fish, and marine turtles. Megafauna, such as marine turtle with a highly mobile behavior, are particularly vulnerable to its impact. Due to their highly opportunistic foraging strategy, loggerheads are more prone to ingestion, but the quantity of ingestion debris is generally low. Turtle entanglement is recognized as one of the most major sources of turtle mortality in the Mediterranean. Fishery gear, but most notably monofilament and micro-multifilament lines and net, as documented as the most significant source of the entanglement for sea turtle. In fact, entanglement has the potential to cause a range of fatal and non-fatal impacts because debris or fishery gear cause strangulation of anatomical parts such as flipper or the neck that led to maiming, increasing drag, restricted movement, or choking. In the image on the right we can see spontaneous amputation. Now we call spontaneous amputation when the flipper is lost in the sea before that the animal are referred to us. And of the right flipper and signs of entanglement on the neck of the turtle. The constriction of the flipper tissues causes alteration of vascularization with consequent ischemic areas which may evolve into necrosis. If this constriction is not quickly resolved, it causes deep laceration of soft tissue, but also fracture, bone loss, and bone exposure. Furthermore, secondary infections are always present, and osteomyelitis is often observed, so partially or completely amputated forelimbs are common, and turtles who have difficulty diving cannot rest, cannot eat. Today I want to talk about a paper that has just been accepted by the Journal of Wildlife Disease. The title is Decision Making in Treatment of Entanglement Flipper in Sea Turtles, Assessment of Residual Vascularization of Limb as Prognostic Factor to Avoid Flipper Amputation. For us, is a very important paper. The aim of this study was to evaluate with the color Doppler ultrasound, MDCT, multi-detector computer tomography, any residual vascularization of neovascularization in entangled flipper of 12 loggerheads, monitoring over time the possible revascularization of ischemic flipper areas, even when the sensitivity is absent. When residual vascularization was present, we analyzed which vessels are involved in neoangiogenesis. The final aim of the study was to understand when it's really, really necessary to intervene with the amputation of the entanglement flipper, or when it is possible to treat the lesion with conservative therapy. Just to remember, this is a skeleton of front flipper of a caretta caretta. The flipper that is most frequently involved, in fact, is the front flipper at the humeral level. At this level, the vascularization is provided by the brachial artery that you can see in red and by axillary vein that you can see in blue. The innervation of the anterior flipper is provided on the cranial part by radial nerve and ulnar nerve, and the caudal part by medial nerve and supracorticoid nerve. For the ventral surface, we have that the ulnar nerve, that the ulnar nerve and medial nerve cover the major part. The constriction can lead to a variable degree of ischemia. So we can see a necrotic limb, necrotic flipper, or only a little lesion, but as in the right image. But most of the flipper entanglement by fishing line are entangled by fishing line and nets that often cause fracture when animals try to free themselves. Soft tissue lesions are often associated with an humeral fracture, but we should not be fooled by the external appearance. The appearance of fracture is not always evident, so radiographic examination is mandatory. So for the diagnosis of entanglement after a physical examination, radiographic examination is mandatory, as well as neurological examination. When a vet is in front a turtle with affected limb and with the entanglement and with the necrotic margins, soft tissue lesion, bone loss, bone exposure, and with the reduced or absent sensitivity, he often decide to amputate due to the bad condition of the limb, which is often connected only by a shred of stump of tissue. Amputation for turtles is has a very important impact. Impact for amputation include the loss of normally swimming and diving ability or mobility, and difficulty in eating and reproduction with with the reduced nesting. In this video from Professor Di Bello, from Professor Di Bello in Lampedusa, we can see a turtle without the right front flipper that swim with her left contralateral left front flipper and with the caudal right front flipper, and the left caudal flipper is used for only for balance. So they can move, but don't move as they can with the other flipper. So it's a very problem. Between June 2016 and May 2018, 12 entangled flipper caretta coming from different Italian rescue centers were referred to the sea turtle clinic of our department. The majority of the animals were found either drowning at the sea or stranded on the coast. Upon arrival at the SCTC, we make a complete physical examination in order to assess the health status of each subject and to evaluate the extent of the tissue examination. We make a careful examination of the flipper affected by entanglement and the movement of the affected limb in and out. This is very important, in and out the water, as well as the ability to use the flipper for swimming were evaluated. And we do also blood test, complete blood test, and culture swabs from lesion, but after the curettage. I now explain in how we carried out this kind of sample. Afterward, neurological examination was performed to assess the functionality and the sensitivity of the flipper. The withdrawal reflex and the deep pain, the withdrawal reflex is the retraction reflex, okay? and deep pain were evoked by compressing the flipper margins with hemostatic forceps starting from the cranial margin up to the entire caudal margin. After neurological examination, turtles underwent radiographic examination of all the body to check also for hook, for example, and of the limb to check for fractures. All the turtles were then subjected to ultrasound examination and underwent color Doppler ultrasound. Ultrasound examination were performed using a portable USB device, and a linear multi-frequency probe was used with the longitudinal and transversal scan optimized for anatomical vascularization study of the entanglement flipper. And when present, the contralateral flipper serves as healthy control. Moreover, we use ultrasound to visualize the intestinal canal to check eventual fishing lines ingested by turtle in an attempt to get free themselves. When they try to free themselves, they take the lines with the mouth and then ingest the lines. For ultrographic examination, the turtle were placed in ventral recumbency on a foam mattress. No sedation was necessary, and the animals were manually restrained, and the flipper were extended as needed to reduce turtle stress. The eyes were blinded by applying a self-adhesive elastic bandage. They are selvatic, wild animals. So we try to don't impact too much with this animal. And the body surface was kept wet using a soaked towel. But when better visualization of residual vasculature and assessment of the bone damage were necessary, a total body multi-detector computer tomography was performed with a 16-slice MDCT multi-detector CT scan. And 5 turtles underwent a total body multi-detector CT scan. In all patients, CT images were acquired before and after manual injection of iodinated contrast medium in the external jugular vein through a 22 or 18 gauge venous catheter on the base of the weight of the animal. All the animal in MDCT underwent total anesthesia with propofol, only with propofol for 5 milligrams per kilogram. For the therapy, in case of total absence of vascularization, the therapeutic protocol provided was the amputation of the entanglement flipper. In the case of residual presence of vascularization, even when this was minimal, the flipper was subjected to surgical curettage and daily treatment with 1 primary wound dressing and Vaseline as waterproofing. All the surgical procedure on loggerheads were carried out after general anesthesia with 5 milligram kilogram tramadol intravenously and before 8 milligram kilogram propofol intravenously. Before surgical curettage, precise wound cleansing of the flipper lesion and disinfection of the surgical areas were accomplished, and all the necrotic and infected tissues, including necrotic or loose bone fragment, were removed. Also for the bone, sorry. After surgical curettage, microbiological swabs were taken, sempre when inoculated on Columbia, blah, blah. Adriana said much better than me, the protocols. And after we used, following the surgical curettage, we used 1 primary wound dressing that is primary wound dressing that is a new, not so new now, is a medicament that is formulated from a synergically combination of neem oil and St. George wort oil. iperico in Italian. And is a good medicament because it's an all-in-one treatment for traumatic injuries and chronic injuries that we use very, very much on sea turtle. After one bet, we use, as said previously, Vaseline as waterproof. Every day before the application of 1 PWD, the wounds were cleaned with gauze and forceps. In this video, there are students that every day works on the sea turtle. Turtles subjected to curettage were not treated with antibiotics. This is, for me, the most important things. And tramadol was used as an analgesic for the first 2 weeks. This is another important thing because turtles suffer, turtles feel pain, so we have to reduce this pain as much as possible. Object of this study were 11 turtles with entangled anterior flipper, Arancia, Calabria, Reol, we know, and one turtle, both caudal flipper, with the absence of the right flipper, with spontaneous amputation of the right flipper. In these 12 turtles, we saw 14 entangled flipper, 4 turtles right from flipper, 4 turtles left from flipper, 3 turtles both from flipper, 1 turtle both caudal flipper. Curved carapace length notch to tip ranged from 22 to 21 centimeters. Weight was 1.35 to 35 kilogram. I said this because our turtles are juvenile stage. In juvenile stage, and this is important because in literature said that entanglement is especially in juvenile stage. We have only 2 subadults with entanglement. 10 out of 12 loggerheads were in pure state of nutrition. You can see the plastron, very faucet, with and poor fat in the axillary and inguinal fossa. All the lesions were very severe with deep laceration and loss of soft tissue, bone exposure, necrosis, and severe secondary infection. In all 11 turtles with the coronal flipper entanglement, the constriction was at a humerus level. The case of the monkey was very severe because the turtles had bilateral involvement with the left flipper lost and only a stump of the right flipper. A total of 7 different gram-negative bacteria were isolated from deep laceration of soft tissue of the entangled flipper obviously after curettage, because before we take everything that is in the water. So after the curettage, we take the swabs. And absolute exhibit resistance to beta-lactam antibiotics, including imipenem, followed by tetracycline and quinolones. 2 strains were found, as Adriana said before, to be resistant to all the antibiotics. In this video, we can see the neurological examination of the flipper monkey that didn't show any retraction, or reflex or deep pain of the stump, but show reaction to the pinching of the axillary skin. At the neurological examination for 9 from flipper, the withdrawal reflex and deep pain were absent on the caudal or all on only the proximal part and distal margin, whilst for the 4 front flipper, the withdrawal reflex and deep pain were completely lost. As you can see, this is vis. The caudal margin didn't show any retraction reflex, any withdrawal reflex. Also the caudal margins. but the turtles show, as we can see just a while, in the cranial margin retraction reflex. So the innervation was conserved only on the cranial margin, not in the caudal distal margin. The entangled caudal flipper, Petrolina, did not show withdrawal reflex or deep pain along all the margin, although minimal sensitivity was maintained on the ventral surface. One flipper, for the radiographic examination, one flipper showed no fracture, 10 flippers showed humeral fracture with severe bone loss. One turtle showed also radio and ulnar fracture, 3 turtles showed the absence of metacarpal bones, and 1 turtle showed femoral fractures with severe bone loss. At the color Doppler ultrasound, the flow of the brachial artery and part of the radial artery was clearly visible. Flowing small subcutaneous vessels were still visible proximally to the constriction of those flippers. The flow in the ulnar artery and the caudal margin, where we don't see any reaction, were not identified. Distally to the flipper, there was no flow. In all 10 cases, ultrasound showed severe edema of the caudal margin of the flipper. A bicep muscle and extensor carpus were completely dissected, and 2 turtles didn't show any flow in all the flipper. After 1 month, the ultrasound follow-up highlighted the presence of angiogenesis of superficial collateral vessels in the residual muscles and of subcutaneous tissue in all the 12 entangled flipper. You can see before and after. In addition to external entanglement, 2 loggerheads had sonographic finding of this intestinal liner foreign body. We find the fishing line because when we find with the ultrasonographic investigation signs of liner foreign body and are every time fishing lines, we perform also a celiotomy to remove this and enterotomy to remove this fishing line, of course. In all entangled anterior flippers, CT show variable degree of bone damage of the middle part of the humeral diaphysis at the level of subcutaneous narrowing. You can see in these images, and ranging from an incomplete fracture, from an complete fracture with multifragmentar, of even complete loss of bone in the diaphysis of the humerus. For the MDCT vascularization is like an angiography, MDCT angiography. In all patients, when the contralateral flipper was present, the vasculature was clearly accessible from the radial ulnar artery to the brachial artery to the subclavian artery until the brachiocephalic trunk. In all patients with severe narrowing of the brachial segment of the flipper and humeral fracture, the main trunk of the brachial artery was interrupted, as we can see in these images. And to also appear connected in some case by small tortuous collateral vessels. 2 patterns of collateral circulation were evident. One deep, large collateral surrounding and bypassing the bone fracture with an evident perihumeral course. If you can see around the bone, there is a large and deep collateral surrounding and bypassing the fracture. And the second one was a small, thin collateral. You can see here. Small collateral that bypass the constriction. In entangled posterior flipper, it was possible to observe bone damage and bone loss of the middle part of the femoral diaphysis with complete fracture, but the main trunk of the femoral and the sciatic arteries were present. And after 1 month, the CT follow-up highlighted the presence of new collateral, mainly in the sciatic artery. 2 turtles with a total absence of vascularization were amputated. This is Iside, and this is Iside after 40 days the surgery. But 12 flipper in 10 turtles with residual vascularization at imaging underwent surgical curettage and treatment with 1-PWD and Vaseline, and the soft tissue